So as previously mentioned, glands are epithelium derived, not just your sweat glands and things like that, but endocrine glands, which is hormones, glands that release hormones, are derived from epithelium as well. We won't worry too much about our endocrine glands just yet. Exocrine glands are glands that possess ducts. So the cells of exocrine glands are going to secrete some kind of product. This one's going to be using exocytosis to get some cellular product out into this lumen, out into this space, out into this duct. So this is going to be the general structure of an exocrine gland, where you have these many arrangements, sort of like these, almost like grape-like arrangements, where you have the cells producing the secretory product, and they're producing it into a duct. That is our definition of exocrine gland. All right, that's really it. That's all you need to know for that. On to connective tissue. Connective tissue is another relatively large category. It doesn't have quite as many of options as we had for naming conventions for epithelia. It's a slightly lower number. There is still some pattern that you can use, but some of our linguistic categories aren't as linear as this, sort of flow chart that we have for epithelia. So most of what you are is connective tissue. Connective tissue is like the packing peanuts. It's glue, it's filler, and occasionally you'll get something in this category that just doesn't fit anywhere else, like blood. Blood is not filler. Obviously there's a lot of functionality to blood beyond it taking up space, but that it just doesn't belong in any other category. It's not a muscle, it's not a nerve, and it's not lining anything, so we just shove it in the category of connective tissue. So most of our connective tissue is going to fit into this, just taking up space, making us into a human shape category. And a lot of dissection is just getting rid of connective tissue to see the other types of tissue that you're looking for. So cells of the connective tissue, different cells for different types of connective tissue. We're going to see a lot of fibroblasts in our general connective tissues, and then when we get into very specifics, bone is also considered a connective tissue, so cells of the bone, osteocytes. Fat is a type of connective tissue, and we're going to call that adipose. You're going to like adipose. It's very easy to recognize. So we have a sort of general structure for connective tissue, and that's going to include a lot of collagen, elastin, reticular fibers, and what we call ground substance, which is a mixture of these things and water, carbohydrates, like complex chains of carbohydrates, which are going to help hold water in, and various things like salt. So we call that extracellular matrix. So here's a cell, for example, of your connective tissue. And one of the things that categorizes connective tissue for most of them is that the cells are secreting or creating or synthesizing the ground substance, the extracellular matrix. The stuff outside of the cell is created by the cells. So we're going to call a lot of these things fibroblasts, for example. Here's a fibroblast. So fibro for fiber, blast for builder. That's what those words mean. So this cell is a fiber builder, and it's actually going to be secreting maybe collagen, maybe elastin. Here's an elastic fiber. Here's a thicker collagen fiber, for example. I think it's got some reticular fibers maybe over here. No, maybe not. So this is just a prototypical organization of connective tissue. There's no one connective tissue that's going to look exactly like this. We're just kind of showing you the types of cells that can be found there. You can also see some white blood cells because we need to have an immune system that reaches all parts of our body. So these will tend to be mobile in connective tissues. Blood vessels, these are vascular. So remember, epithelia are avascular. They do not have blood supply. But connective tissue, by definition, is vascular. You will have a blood supply within connective tissues. And here's some fat cells. Adipocytes, they tend to clump together, and you'll find them in a variety of locations. And you'll generally be happy to see them. You'll be like, oh, it's my old friend adipocytes. I can recognize him. Any Doctor Who fans? Just you, so you know about adipose. Yeah, they're so cute. I'll show you guys later what we mean. Okay. So connective tissue, it's physical protection. Even just those layers of fat can be protective. It's a cushion. Suspension, support structural framework. Your eyes are actually embedded in a whole bunch of fat that just keeps them in place in your eye socket, in your orbit. Binding structures, storage. Again, adipocytes are great for storing those fat molecules for when we need energy. Transport and immune protection. We do have, again, those macrophages. Macrophage, does anybody want to break that down for me linguistically? Macro is big. Phage. What's that? Looks like Phage. 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 Phagocytosis was eating. Yep. So it's a big eater. So if you've got a bacterium hanging out over here, this macrophage is going to come over and it's going to engulf it, phagocytosis, and it's going to destroy it. Okay, embryology. These words probably don't make sense right now. So embryologically, you started out as 3 pancakes. An outer pancake, a middle pancake, and a lower pancake. And they had names. They were this is the wrong color. We color code them in the sciences because we're fun like that. I'm going to use, let's see, do we do blue for inside or outside? Yeah, we blue inside. So here's the lower pancake. Here's the middle pancake. Here's the outer pancake. This is yellow, but you can't see yellow, so it's an orange. That middle layer becomes your muscles, it becomes your skeleton, and it's telling you here that this part of this middle pancake, the mesodermal mesenchyme, is going to become most of your connective tissue. So this is the middle, becomes the middle stuff. This is going to become your gut tube. This is your endoderm. Your ectoderm becomes your skin and nervous system, and your mesoderm, which has mesenchyme, becomes your connective tissue. So think of this as like a structural framework for everything else. And that's really all that we need to do. So again, the keywords that you need to know are on the slides. They're not necessarily things that I throw at you sideways. I know we've got a lot of people interested in OB, obstetrics, and labor and things like that in here. So I'm doing a little bit more with the embryology. Obviously, it takes a lot to get from 3 pancakes into something that looks like a human. Quickly, soon after this, this pancake, this wide flat thing, curls in on itself laterally and from the cranial and caudal ends and gets into that sort of bean-shaped fetus shape. It's kind of cool. All right, so there's the middle stuff. That's what they mean by mesenchyme. Any questions before I move forward? Okay, we can hit those lights if somebody's mobile enough. I don't think I emphasize embryonic connective tissue. I don't know that this image is in the images that I gave you necessarily. Okay, I'm not going to. So classifying connective tissue, there's connective tissue proper, and this does have a naming convention similar to like we had for epithelia that makes it easy to intuitively arrive at the name. But you also have supporting connective tissue, and fluid connective tissue is just blood, so that's a small enough category. Supportive connective tissue has its own discrete pattern that we're going to use for it. Again, we're going to emphasize patterns anytime we can, because if you can find the patterns, you can cut down your studying. So connective tissue proper, we're going to have either loose or dense, and we're going to have either regular or irregular. So that's where we can do our sort of flow chart. Is this connective tissue dense? Is there a lot of white space? Is there a little white space? And then you can say, is it regular or irregular? Are these fibers, are these cells oriented in the same direction, or are they all going in completely random directions? With a couple of exceptions for those naming conventions, it's not as clear-cut for naming conventions as it was in epithelia, unfortunately. Supporting connective tissue is going to be either cartilage or bone. And we put these together, we're going to learn later this semester that cartilage becomes bone, so they're very closely related structurally and by location as well. You're going to have 3 types of cartilage that you're going to need to be able to recognize. Bone, you're going to love bone. It's so easy. It looks like a tree trunk. And then fluid connective tissue is just blood. The only type of fluid connective tissue we have is blood. It's kind of weird to put blood in this category to start with, because the fluid of your bloodstream is not being secreted by the cells of your bloodstream. Okay. Yeah, I think I don't want to go into that. Collagen fibers are going to be a very abundant protein in connective tissue. It's going to be strong but flexible and resist stretch. So your skin goes like this, right? Underneath your epidermis, underneath your dermal tissue, underneath those layers you have a lot of connective tissue layers, and the collagen under it, it doesn't aid with the stretching bit, it aids in the solidity of it. I can't peel off my skin, thankfully. Not just because of those desmosomes in my skin, but also because of things like collagen fibers and some very sturdy connective tissue under it. So that's a good thing. I mean, I could if I really tried, but let's not try. It's a very abundant protein. Elastic fibers are going to help with the stretch. So the fact that I can pick up my skin like this and go like that without really hurting myself, without peeling anything away, that's in part due to the elastic fibers. And then reticular is going to be a little bit of an intermediate. Certain organs are going to have reticular fibers. If you see reticular fibers, you're in specific places that are going to help kind of with filtration. These are all filtration organs, is one way to think of that. These are each going to stain a little bit differently, and sometimes we need to really stain for that fiber. It's going to be hard to recognize an elastic cartilage, for example, unless you've stained for an elastic cartilage. Luckily, our elastic cartilage is stained for elastic cartilage, so you'll be able to identify the elastic fibers. Okay, I covered that. As I said before, loose versus dense, regular versus irregular is going to be our naming convention for this. So let's dive into it. Areolar connective tissue is going to be another name for loose, irregular connective tissue. So you look at this and you see some white space and you see some random orientation of fibers. Do you guys agree with me on that? Lots of white space, random orientation of fibers. That means that you are definitely in a loose, irregular connective tissue, and the phrase for that is areolar. Frankly, if you write loose, irregular connective tissue, I will be pleased as punch. I'm okay with that. Technically, the term for it is areolar. They do have elastic fibers that they have stained for. They do have collagen fibers that they have stained for. That's what's going to be found immediately below the skin. That is what enables us to pick up our skin and use some calipers on it if we want to. Adipose is awesome. Adipose is going to look like this big fluffy cloud right here. So anytime you have an adipose cell, you're going to have a big fat globule taking up most of the space of the cytoplasm, And you'll just see a tiny little purple dot. That's the nucleus for this cell. That's the nucleus for, oh no, it's over here. Look at the tiny little nucleus compared to this gigantic fat globule. Isn't that adorable? Look at it. You just want to squish it. It's going to be really easy to spot in tissues because suddenly you're going to see this big old white space. It's not an epithelium white space, it's a fatty white space. It's going to be completely encircled. It's just going to be little spheres of white that show up in histology. Okay, reticular connective tissue, we had to stain for these reticular fibers. It looks a little gnarly to me. The spleen is a filtering organ, so if you want to sort of think of this as a net that's catching stuff, that's one way to start thinking about that. Sometimes when we stain for these darker fibers, it looks kind of hairy to me. It kind of looks like a clogged sink a little bit. Any questions about those so far? So again, this is, you could argue, loose and irregular connective tissue, a form of loose irregular. We've got a lot of white space and we've got a lot of random orientation of the fibers in it. Do you have a question?
Yes, so if this was like on our lab vacuum, what would we have to label?
Reticular connective tissue, for sure.
But like,
it would just be ID this
or just
tissue.
the whole thing in general, point to something specific.
Again, most of it's going to be ID this tissue. I think the only specific thing I could potentially ask you is where would it be found, and since it is called reticular connective tissue, I could maybe ask you what type of fibers those darkly stained fibers are, and you would say reticular fibers. And hopefully, I mean, I would try to make it be something that obvious that, like, if you get it, then you've already got it. Okay. Here's our first dense tissue. This is dense, and it's irregular. So here's your purple dot. There's your nucleus. And this is wavy, so this is actually in a tendon. This is going to be a relaxed tendon. When this tendon is stretched out, it's going to be perfectly straight. But since this is a fixed specimen, then by definition it's dead and relaxed. So it's wavy. But the orientation of these collagen fibers is all in the same direction when you stretch it out, right? It's all going the same way. So we don't have anything going this way at all. It's all going in the same direction. And there's not a lot of white space, so you can definitely say, okay, this is definitely a dense connective tissue. I think this is the easier of the dense connective tissues to identify. It is possible to confuse this with a completely different type of tissue. I will point it out later. Our images specifically make it pretty clear. If I would throw left field pictures and pictures from different sources, it would be easy to mix this up with some smooth muscle that we'll see later, but we're not going to do that to you. We're only using this picture. This picture can be a little challenging because this is considered dense. What did I tell you about identifying dense versus loose? A lot of white space. All right, so this kind of looks like a lot of white space. What I want you to do is compare this white space to that white space. Which one has more white space? The areolar. In my opinion, the areolar has more white space. So one thing that happens when we fix histology is that sometimes these things get broken apart, and there wouldn't be as much white space in the living specimen as there is in this. So they've kind of torn apart in the process of fixing this histology slide. It's called an artifact of histology. So you kind of have to mentally compress this image. I know it's not intuitive that you'll have to do that. Again, you are simply rote memorizing this image, so that makes it easier. The idea is there's less white space, so it is dense. All of these fibers would be interacting directly with each other, but it is randomly oriented, so that part is relatively easy. This is going to be even deeper under your skin. So we start with some loose connective, some areolar connective tissue close to the surface of your skin, and then as we get deeper, it becomes reticular. It becomes, sorry, I shouldn't say that. That becomes dense irregular connective tissue. Here's another dense and irregular type, but it is stained very darkly and completely filled with elastic tissue. So we're going to call this elastic connective tissue, even though it's fairly irregular. It's a little irregular, and it is very dense. We're going to call it elastic connective tissue because of the elastic fibers. Okay, supporting connective tissue, cartilage, and bone. Bone is another one that it does fit fairly well into the connective tissue orientation because the cells of the bone are creating the bone matrix. It's just at one end of the spectrum. At the hardest end of the spectrum for connective tissue, you have bone. At the fluid end of the connective tissue spectrum, we have blood, right? They're the two extremes. Cartilage is going to be in the middle there. So again, we won't use our naming conventions for cartilage. It's not dense, it's not loose, it's not regular or irregular, it is cartilage. But it's still connective tissue because the chondrocytes, the cells of the cartilage, are synthesizing the extracellular matrix. The ground substance of cartilage is made by chondrocytes. One thing that I'm a huge fan of, I'm going to dance around this and shake my hands a bit about this, both chondrocytes and osteocytes, so cells of the cartilage and cells of bone, they make little houses for themselves. They make little houses called lacunae, and I just think it's a really cute word. So they have these little houses, these little pockets for the cell to live in as it maintains the space around it, as it maintains the ground substance. So chondrocytes live in lacunae, osteocytes live in lacunae. Lacunae. It's just a cute word, right? Don't you just like, oh, it's got a little house. So luckily there's only 3 types of cartilage. We're already starting to narrow down our numbers. Hyaline cartilage, fibrocartilage, and elastic cartilage. First up is hyaline cartilage. It is the most common. I disagree that it's the weakest. That's wrong. I think your textbook says this, but it is incorrect. Hyaline cartilage, the location is important to you. You are going to see these cartilages again when we get to bones. So I do want you to know some general locations for each of these cartilages. Hyaline cartilage is going to be found at the ends of long bones. It's going to be in your articulations. Within your joints, you're going to have hyaline cartilage. So here are your chondrocytes within their lacunae. Look at them all happy in their little hidey holes. Look how cute they are. Oh. And the extracellular matrix is the cartilage, and it has a glassy appearance. It may be hard to see it just from this one slide, but if you're looking through a light microscope, it looks kind of like a stained glass window. It's really lovely. I'm actually going to compare and contrast this first to elastic cartilage. If we had not stained for that elastin, this would pretty much look just like hyaline cartilage. You still have chondrocytes within lacunae, and you still actually do have a glassy extracellular matrix. The difference is the presence of abundant elastic fibers, and when we stain for those elastic fibers, then we can clearly see that this is elastic cartilage. So it's that sort of hair-like texture that when you stain that elastin dark like this. Yes?
What is the purpose of the lacunae?
So, I mean, they're literally housing. It's a space for that cell to be, and once that cell is embedded in that space, its job is to maintain the extracellular matrix in the immediate area. Yeah. Yeah, I'm not being cute and adorable. It's being cute and adorable by having a little house. Yeah. So
Those are the lacunae?
yeah, so the big spaces, and then you can see the purple dots within the spaces, at least in the medical illustration version, you can see the nuclei of the cells that are inhabiting those spaces. Yes, so the blobs are the lacunae. Okay, so again, hyaline cartilage, they have the lacunae, they have the glassy extracellular matrix. The elastic cartilage additionally has the elastic fiber that has to be stained for. You will see elastic cartilage in a couple of very important places. Your ear, so the fact that your ear goes like this and that doesn't hurt, that's because it's elastic. The other really important place is going to be your epiglottis, and that's the thing that covers your trachea when you eat food or swallow your coffee. So we have to cover that air tube, that trachea, with some elastic cartilage to keep fluid from going into your lungs, to keep food from going into your lungs. I consider this to be the weakest one, not hyaline cartilage. I consider this to be weak because that elastic fiber does wear out over time. The elastic, as you age, your cells become less capable of creating these substances out here, and that does wear down, and there are real consequences for that. So the elderly are at an increased risk of choking. Have you guys heard that? It's because their epiglottis can wear down because of those elastic fibers wearing down.
Like in that example that it says external ear,
Mm-hmm.
as we age, what can happen to the ear?
I mean, unless you're going like this all the time, probably not much. But if you went like this, then probably have a weird saggy ear by the time you were elderly. Yeah. But it doesn't face a lot of stresses.
So that if that is held in the epiglottis, the external ear, and the trachea?
So the epiglottis is at the mouth of the trachea. So that is the same location. For a lot of these, if you want to develop pattern recognition skills, Google image search is perfect. If you type in hyaline cartilage, you will find 50 different pictures of hyaline cartilage in different locations throughout the body, different stains for hyaline cartilage. That is one good way to develop pattern recognition without, spending a lot of money, without needing a lot of resources. It's an easy. Do you have a question? Or are you stretching? Okay, just stretching. It's a really easy resource for most of them. The exception is fibrocartilage. Every time I look up fibrocartilage, it looks like 25 different things to me, and I don't know why. So everything except for fibrocartilage, go ahead and develop that pattern recognition with Google image search. Don't do it for fibrocartilage. We've got a weird blue stain here. I don't know why we have a weird blue stain. Nobody else has this stain. I do agree with the way that they've organized this. There are still chondrocytes and lacunae, and that's identifiable for cartilage. And then you have these dense collagen fibers, kind of bundled and wavy in this orientation. I agree with that. I think that's perfectly acceptable for a representation of fibrocartilage. Again, if you do a Google image search for it, you'll find 16 different ways that this is supposed to look. So again, the location for this will be significant to you. You will find these in very, very solid joints like the intervertebral discs. We'll learn in our joints lecture that you have these disc-shaped articulations, and they are a special category known as a symphysis for a joint. So I do think that fibrocartilage and its existence within the intervertebral discs is significant to you long term. Okay, question about the 3 types of cartilage. Okay. Bone is actually formed from cartilage, so there's going to be that structural relationship between them. It has its own appearance. We actually don't need to stain bone to get this appearance. To me, it looks like we clear-cut a forest and we're looking down on it. These look like tree trunks to me. So I think this is very, very easy to recognize in every context. It's one of my favorites for looking at. Somebody hands you a random histology sample, you hold it up and it looks like tree trunks, it's bone. We have to look closely to see the lacunae. But what's happening with these tree trunks is the bony matrix was created in circumferential rings, very much like tree trunks were. It's not coincidence, it's evolution. So we formed sort of from the inside out, and as we made it, we created lacunae, and the osteocytes live within the lacunae. So within the rings, we have these black dots now. This is not stained, so they don't look purple, they look black. Each one of these little dots right here, that's an osteocyte within a lacuna. So you're looking within the circumferential rings for those black spots. Those are osteocytes within lacuna now. So they don't look quite like these lacunae. They're more flattened, and they're interspersed amongst the rings of these osteons is what they're called. But we'll learn about osteons when we get to our osteo unit. Okay. Whee! Again, other end of the spectrum, this was the solid end of the spectrum. Those osteocytes do maintain that sort of calcium phosphate mix of the bone, the bony matrix. That is the solid end of connective tissue. This is the fluid end of connective tissue. The difference here is, again, these cells do not secrete their extracellular matrix. The extracellular matrix here is pretty much just fluid, electrolytes and proteins. So we have red and white blood cells and platelets, and that's it for cellularity here. Those are the only cells you're going to have. Platelets are cell fragments, so that's not even a cell. The watery ground substance is known as plasma. Red blood cells you can also call erythrocytes. The prefix erythro means red. White blood cells you can also call leukocytes. And cell fragments known as platelets also circulate around. They participate in clotting. Okay, what's this type of cell? Red blood cell. What's this type of cell? White blood cell. What's this thing up here? Platelet. Oh. All right, that was it for connective tissue. So significantly shorter than epithelia. And guess what? Muscles, we've got 3. Nervous, we've got 1. So we're almost done already. We've got 10 slides to go. How are you guys feeling about this so far? It's not too bad. Hopefully it's not too bad. Okay, 3 types of muscle: skeletal, cardiac, and smooth. Not surprisingly, this concept is going to carry forward because we've got an entire unit. Unit 3 is on muscles. So for categorizing muscles, there's the visual categorization, and then there's a little bit functional. What I'm going to want you to know about is it voluntary or voluntary? What is its appearance? How many nuclei does it have? Standard cell has 1 nucleus. Some of these cells are going to have more than 1 nucleus. They're a little weird. And if there's anything cool about it. Honestly, I think there's something cool about each and every one of them. The most important cool feature is going to be on the cardiac muscle cells. So skeletal muscles tissue is going to be voluntary. We decide to move. I gesture like a cool person, which I obviously am. And that was a voluntary movement created by skeletal muscle. The appearance is striated, which means it's stripy. By the end of the semester, you'll understand why those stripes are there. For now, we're just going to roll with the idea that they are stripy. It's due to the organization of the cytoskeleton that allows for contraction. It's going to be very linear, and that's going to make stripes. These will be multinucleated. Skeletal muscle cells pretty much always have more than 1 nucleus just in one very, very long cell. They're going to be long cylindrical fibers. They're going to be striated. They're going to lie down in a parallel orientation. This is kind of a weird slide for them to have chosen. It's very, very dark. I think it's just the balance has been shifted. I think they took the white balance and manipulated to show you very clearly how stripy it is. So it's okay. It's nice that it's linear as well. It's an acceptable image to use. I don't know why they went so hog wild with the contrast, but it's on them. Cardiac muscle is also going to be striated, so we're going to have to use another feature of cardiac muscle tissue to differentiate it from skeletal muscle. Now, do you have to think about your heart beating? Do you decide for it to beat? No, it is involuntary, so completely subconsciously controlled. You don't decide to do anything with it. If you did, you'd probably die. These are usually only with one nucleus, usually uninucleated. They tend to have branched, Y-shaped, and shorter cells. So the cells are not these really long cylindrical cells. This is a very high magnification. The one we're going to use for cardiac is a little bit lower magnification. So we've zoomed out a little bit. You can still see some striations, especially in the medical illustration version. If you look carefully at this image, you can see that there are striations. I think this part has some fairly clear striations. Again, this is on a different scale than this one. You can very clearly see the scale of the striations on this one compared to the scale of the striations there. This is zoomed out slightly. But the one thing that's really going to make this clear to you that this is cardiac muscle and not skeletal muscle is the presence of intercalated discs. Intercalated discs are a site of gap junctions. And this is one of those things where I'm going to dance around and shake my hands a bit and say gap junctions are a concept that never goes away. AP2, you've got a whole cardiac unit where we're going to talk about how those gap junctions allow for communication from one cardiomyocyte to the next, cardiac muscle cell to the next, because they need to share ions. When an electrical signal arrives at this cardiac muscle cell, it needs to get to this cardiac muscle cell immediately, because your whole heart has to beat in a coordinated fashion. All of this tissue is going to contract at the same time, and then all of this tissue is going to contract at the same time. And it cannot happen any other way. If it does, we are sick. So the intercalated discs with gap junctions facilitate that cell-to-cell communication, that instantaneous communication of contract now. Contract now, contract now, contract now, contract now. Yes, Claudia?
On that note, what about what does it have to do with the muscle tissue, maybe like arrhythmia? Because that's
It's exactly what we're getting at. So an arrhythmia is discoordinated contraction of muscle cells, and generally that's going to be about a problem communicating between cells. Usually it's going to be some amount of heart tissue has died. That's not the only reason that could happen, but that is one major reason that those happen.
That leads to potential heart attack.
Yes, arrhythmias can lead to a heart attack and vice versa. Yeah. Yep. That is exactly why we emphasize this. Okay, last muscle tissue is smooth muscle tissue. It's going to be found in most of your internal organs. It's going to be found in your blood vessels. So do you have to think about your intestines churning away at your food? No, you absolutely don't. So this is involuntary. They are going to have one nucleus, uninucleated. They are not going to be striated anymore. So you're going to need another visual identifier to tell you that this is actually muscular at all. So smooth muscle, they're fusiform. They're going to be a sort of wide, flat shape. They're short, they are non-striated. They still have the cellular mechanisms for contraction, obviously. But whereas the striated ones, they were all arranged in a linear fashion, side by side. We're all going to contract in this exact same orientation. Smooth muscle cells have those units in a randomized orientation. So when it contracts, it pulls in from all angles all together, meaning that it's not going to have a striated appearance. We're also going to have a sort of flattened nucleus. Take a moment, look at this image. What do you think you would mix this up with? What's that?
Dense.
So yeah, dense regular connective tissue is the number one thing. There's actually others that we're not even doing. Peripheral nerves can look like this. If you forgot about the white space rule for epithelium, you might think of maybe a stratified squamous epithelium, because fusiform is squamous, more or less. We're just using a different term since it's a different type of tissue. So this is very easy to confuse with others, which is another reason I'm only using this one slide for this one tissue. I'm not throwing you any curveballs. Okay, that's it for muscles. We already finished. It's super easy. And then there's just one more, nervous tissue. 2 types of cells in nervous tissue. If I had to come up with a whole bunch of test questions on this, I might, go into identifying those individual cells. Hint, hint. So nervous tissue, you're going to have neurons and you're going to have glia. And neurons are going to be gigantic and glia are going to be tiny. And that's how you'll know that this is your neuron and this is your glia. That's a glia, that's a glial cell, that's a glial cell, that's a glial cell. So again, you're just looking for purple dots. You're looking for dark purple dots, and those are cell bodies, those are nuclei. These are all nuclei of glia, and this is your nucleus of your neuron. So neurons are going to have a lot of cellular processes, generally several dendrites and one axon. This isn't my favorite picture of a neuron because it's not really clear from this picture, unless you're an expert, which of these are dendrites and which one is an axon. Like, honestly, if you just gave this to me, I wouldn't know if that was the axon or that was the axon or that was the axon. So this is not my favorite picture ever. You can see another neuron right over here too. Cool. The thing about staining neurons, we're actually staining like 1 in 1,000 neurons with this stain. So it's actually much more dense than that, secretly. All right, those are your tissues. With age or stress or changes, tissues can convert and change from one type to another. If you sit on your couch all day, or if you get nerve damage and can't use your legs, if something loses innervation or is simply disused, your tissues may shrink by either cell size or by number of cells that you have. That's atrophy. Those words mean A as in without, and trophy, without feeding. Trophic is feeding. Neoplasia. Does anybody know what those word roots mean? What's neo is new. What about plasia? Growth. So new growth that means there's a tumor. Tumor doesn't necessarily mean cancer. Cancer means malignant. So if a tumor was malignant, then it would be cancerous, but not all tumors are cancerous. Neoplasia just means new growth. It's not growth that we planned for. It is cancer or a tumor. There's a difference between hypertrophy and hyperplasia that I'm going to emphasize. Hypertrophy is an increase in the size of existing cells, whereas hyperplasia is an increase in the number of existing cells. So in adults, some tissues can undergo mitosis, some tissues cannot undergo mitosis. I'm going to hook this up to this idea right here. Remember how our skeletal muscle fibers, our skeletal muscle cells have more than one nucleus? That's because as they grow in size. Gesundheit. They grow in size. They do not grow in number because skeletal muscle tissue does not undergo mitosis in the adult. Skeletal muscle tissue hypertrophies. It will increase in size. When it increases its size, it will actually create new nuclei within the same cell. So that nucleus can serve as, the manager, the guy that distributes the blueprints for each region. Think of it as like regional managers if you want to. So skeletal muscle cell is an example of a place where you would find hypertrophy, as in increased feeding, high feeding, an increase in the size of existing cells, whereas hyperplasia is going to be an increase in the number of cells. So in a place like your skin, where you can have a lot of mitosis, that can increase in number, and then you would get a tumor of your skin from something like that. Or an increase to stress or increase to use. You could have that as a normal thing as well.
So hypertrophy is in the muscle, hyperplasia in the skin.
Yeah, those are good examples. That's not the only situations in which those would happen. They are good examples of where they would happen. Metaplasia means converting one cell type to another. So today we learned about the purpose of stratified squamous epithelium being protective, right? If there's going to be friction there, we want there to be stratified squamous epithelium. In your esophagus, you have stratified squamous epithelium, because if you swallow a Dorito, you don't want that to slice into that skin. You want that to be protected. One thing that can happen in that esophagus is if you have reflux, if you have acid reflux, then that skin can convert from one type to another. It can actually change into the type of epithelium you have inside of your stomach. The inside of your stomach has an epithelium specifically designed to deal with stomach acid, right? It's that it evolved to be safe for stomach acid exposure. So if you've changed your esophagus epithelium from stratified squamous to simple columnar, like you have in your stomach, you no longer have the protection against physical abrasion. You do now have protection against stomach acid. It was something that you adapted to, right? But we will consider that a precancerous condition. So all of these are bad changes, or they can be bad changes, I should say. Obviously, hypertrophy, if you're lifting weights, that's a good thing. There can be hypertrophy that is bad, like if your heart is stressed out and it has to work harder, it will grow, and we call that cardiomegaly, and it's not a good thing. So all of these can be bad. Metaplasia we will consider precancerous. Hypertrophy has its own dangers. Hyperplasia could be the sign of a tumor. Neoplasia is a tumor. Atrophy means it's disused or it is deinnervated. You've damaged your nerves. All right, any questions? That is the end of the slideshow. Yay. Okay, I'll go ahead and stop that.